Good morning, everyone. Thank you for joining us today. Today, we'll be talking about the nervous tissue histology. Thank you for everyone at the peer-assisted learning for arranging this. Without further ado, let's get started. So my main goals with this talk is to get you to understand the orientation of the nervous system and create this sort of big picture understanding of how the nervous system is organized. And then my second goal is to help you understand how questions are usually asked when it comes to histology. And then I'm going to highlight some high-yield points that you need to take away before you enter your exam. I'll keep my talk brief so that you have time to revise and look at further resources. So the nervous system. The nervous system can be simply divided into the central nervous system and the peripheral nervous system. From the name, central is along the central axis of the human body, and it includes the brain and the spinal cord. And then you have your peripheral nervous system, which is all these nerves over here. All these nerves, as you might have learned, the brachial plexus probably by now. And then you have your intercostal nerves here. You have your femoral nerve, lateral cutaneous nerves. So these are all the peripheral nerves, okay? And so as long as you memorize that, the brain and the spinal cord are central, everything else is peripheral. Okay, and this nervous system is made of two different categories of cells, the neurons and your glial cells. Your neurons are the first thing you usually think of when you think about the nervous system, and they're responsible for carrying stimuli or action potential throughout the nervous system. And then you have your supporting cells, or these are called glial cells, and these help the neurons do their function. And they're associated with multiple different processes, which we'll talk about later on this talk. Okay, so your neurons can be divided into sensory neurons and motor neurons. Sensory neurons usually carry stimuli, whether light or sensation or pain stimulus, from the periphery to the brain. So receptor to brain. And then your motor carries the certain function to the muscle. Okay, so it's usually a brain to muscle connection. Now, the glial cells have many different functions. We'll talk about the different cells and each of their functions. But the main theme of these functions are that they protect the neuron. Okay, they're responsible for the defense, and they help bring on neural nutrition. So help the neuron in its metabolism. And finally, they're responsible for the immunity of these cells. We recently published a paper about their role in spinal regeneration, so that helps you understand further that the critical role that the glial cells play. All right, so neurons can be divided into different parts. So you have your cell body, or also known as the perikaryon, and this usually contains the nucleus, as you can see here, the nucleolus, which is this purple, dark purple spot, and then your cytoplasm, obviously, and other organelles. The general way by which a neuron works is by creating products in the cell body and then transmitting these products along the axon to the axon terminal. What these products are usually neurotransmitters, and we'll talk about this throughout this slide. Okay, so the dendrites is where the neurons, it's these tree-like structures around the cell body that receive stimulus from other neurons. And then the axon is what carries the products that were created in the cell body to the axon terminal and deliver it to other neurons or muscles in some scenarios. So you end up with this complex network in which different neurons are connected to each other. And this is how you get to think, this is how you get to create memory, this is how you get to move your hands to see. And it's really critical that this happens in order to carry on basic life functions. So there are different classifications of neurons. This classification is based on the arrangement of axons and cell bodies. So you have your multipolar neurons. From the name multipolar, that means different multiple dendrites are connected to one cell body. And then you have your axon as an output. Then you have your bipolar neuron, in which one cell body branches into two different parts. You have your dendrite and your axon. And then you have your unipolar neuron, in which the cell body is connected to something called the single short process. And then this bifurcates to connect with the peripheral nervous system and then the central nervous system. And then finally, you have your anaxonic An means without anaxonic, so without an axon. So these are usually surrounded by dendrites. Um, it's important to get an idea of the morphology of these cells. However, it's more important to understand where these cells are. So bipolar neurons are usually found in the retina, the olfactory epithelium, and the inner ear. The way you can memorize this is by thinking that these bipolar neurons are responsible for special sensation. The multipolar neurons are usually the most common neurons. They're found everywhere. And then your unipolar neurons, they're found in the peripheral nervous system, that is just the peripheral sensory nervous system. Okay, and I hope I can show you this in the upcoming slides. And then also the anaxonic are found throughout. So I think the main point that you can take away from the slide is that bipolar neurons are usually found in special sensation in the retinal olfactory epithelium and the inner ear. So now let's talk about each and every part of the neuron. So you have your cell body. As I said, this is where you have the most of the cell metabolism happening. Usually you have your DNA in the nucleus. This DNA becomes RNA. RNA becomes protein, and then the protein gets transmitted to the through the axon. The surrounding around the nucleus is made of a complex structure called the rough endoplasmic reticulum. This rough endoplasmic reticulum is responsible for making the proteins necessary for the neuronal function. Now, this structure is basophilic, and in your nervous system, it has a special name, which is the mesophile. Dendrites are a branching structure, tree-like structure around the soma or the cell body, and their main goal is to take stimulus or action potential from the surrounding neurons, as I've shown you before. They are large, and they undergo a process called arborization. Arborization simply means division. So we have something and then it divides into many different things. And this allows a single cell body to be connected to many neurons, up to 200,000 in specific organs. It is also important to note that these dendrites are connected to each other through the dendrite spine. So you have your dendrite, it keeps on splitting until it forms the last part. This part is called the dendrite spine. This is where most of the synaptic connections occur. And then this morphology depends on a protein called actin. This actin helps bring on the shape and the function of the dendrites. We have talked about the structure of the cell body and the dendrites, and now this cell body needs to be connected to the axon here. And this is done by a structure called the axon hillock, and in this structure, it contains many microtubules and neurofilaments. These microtubules, they're long proteins along the axon that carry the products that are made in the cell body throughout the axon to the axon terminal. So as you can imagine, the axon hillock plays an important role. Now, when it comes to the axon, the cell membrane of the axon is called a axolemma, and its content is called an axoplasm. It's important that you get a general understanding and become as oriented as possible to these terms. You don't have to memorize them, just know what they're referring to. Have an idea about what they're referring to. Now, this is another picture to show you the axolemma, as you can see here, and the axoplasm, and this is the microtubule you can see here. Now, this is a good visualization. So as I said, you have your DNA, it makes the RNA, and then this RNA gets translated into a protein, and then this protein gets packaged and carried through by the microtubules along the axon. And then usually most of the time the protein is a neurotransmitter. This neurotransmitter helps the activation this neurotransmitter helps in the activation of other neurons. This is a beautiful visualization of what goes on in the microtubules. You have a protein called kinesin, and it carries the organelles or the vesicles throughout the axon. It's nice to see this in action. There are many videos on the internet, but this protein essentially walks on the axon. And finally, you have your axon terminal or terminal bottom. And this is what happens when the axon undergoes alkalization or division. And what happens that is that these terminals will form the presynaptic cleft. As you can see here, this is a synapse. This is a axon terminal here, and this is a postsynaptic terminal or the dendrite. And what happens here is that the vesicles which were made in the cell body, they diffuse out and they activate this postsynaptic terminal, and then this neuron gets activated, and so on. It's important that you understand these, but I won't dive into the specifics because it turns into a physiology lecture. Now, the different connections throughout the nervous system can be divided into three main divisions. So you have your axosomatic from the name. So it's an axon to a soma, which is the cell body. You have axonic, which is an axon to an axon, as you can see here. And then you have your axodendritic. Again, you don't have to memorize these, just get oriented. Now, this is a synapse. This is the axon terminal. These are the vesicles I was talking about. It is also important to explain what goes on in the synapse. So usually when a neuron is activated, when the neuron membrane is activated, it's going to open these calcium channels. Calcium is going to enter the axon terminal, and then it's going to activate some proteins here, which will allow for the fusion of the vesicles. These vesicles will release the neurotransmitter, and then this neurotransmitter will bind onto the postsynaptic membrane, opening other receptors, in this case sodium channels, activating the other neuron. And this is essentially what happens inside a synapse. So what ends up happening is that you have an electrical signal in the cell membrane of the neuron, and then it gets converted into a chemical signal or a neurotransmitter. And then this neurotransmitter activates the channels on the postsynaptic membrane, and it gets converted to an electrical signal again. These neurotransmitters are many. They include acetylcholine, dopamine, GABA receptors, and each has its own excitatory or inhibitory function. So some neurotransmitter might activate the neuron in front of them, others might deactivate the neurons or inhibit the neurons in front. Now that's for the neurons, their function and their structure. Now let's talk about the glial cells. So the glial cells are many, and they exist to support the neuronal function. Now, as you can imagine, the CNS is only limited to nervous tissue. It does not have any connective tissue. And in order to compromise for this, you have your glial cells. Another term that we can use to refer to the complex arrangement of neurons and glia is called neuropil. It just refers to this matrix of neurons and glial cells together. Just keep that in mind. All right, so these are different glial cells in the nervous system. So you have your oligodendrocytes, your astrocytes, microglia, ependymal cells, Schwann cells, and satellite cells. Each has its own function, and I think it's important that you understand this function before moving on from this lecture. So your oligodendrocytes are these cells here, and their main function is to produce the myelin for the central nervous tissue. In the CNS, you have your brain and your spinal cord. The brain and the spinal cord have areas called gray matter and white matter. I hope I can, yeah. So you have your gray matter and your white matter. In the white matter, you have these neurons that need to carry signals really fast. So these neurons are usually myelinated or have myelin. The beauty of myelin is that it allows the signal to jump from one node of Ranvier to another node of Ranvier, speeding up the process. So this is a node of Ranvier. It's the point which is not myelinated between two points which are myelinated. So it's this point right here. And the signal jumps. So as you can imagine, jumping makes the signal travel faster. And in order to get this myelin, you need specific cells to wrap around the neuron. In this case, if it was the central nervous system, it's the oligodendrocyte. Right, Schwann cells, on the other hand, do the same function, but in a different location. In this case, it's the peripheral nervous system. So they wrap around the axons of the different nerves, such as the femoral nerve, the musculoskeletal nerve, and ulnar nerve, radial nerve. And this helps with the signal to travel faster in those nerves. So unlike the dendrocyte, The Schwann cells usually wrap around one axon, while in the oligodendrocytes, one oligodendrocyte wraps around multiple axons. Keep that in mind. So they differ by location and the number of axons that wrap around. Astrocytes are the most abundant cells in the central nervous system, and we can summarize their function into three main things, three main key functions. The first one is that they form the something called the glial limiting membrane. So as you know, you have your gray matter in the brain. This is your, so as you know, this is your brain, and you have your gray matter, and what forms the final layer, which is in contact with the meninges, is the astrocytes. And I'll show that to you in a different picture. So this is called the glial limiting membrane. And furthermore, they wrap around capillaries and they form the blood-brain barrier. Okay, so they wrap around capillaries and they filter what goes on to the brain. So this is a capillary and this is an astrocyte wrapping around. They create this tight junction to only select specific chemicals or nutrients to pass to the nervous tissue. The microglia plays an important role in the immune regulation of the central nervous system. It does so by removing damaged components or phagocytic functions, exactly like a macrophage. In fact, it does not originate from a neuronal stem cell, but instead it originates from the blood. Ependymal cells form these tight junctions in which CSF or cerebrospinal fluid can flow, and they wrap around the ventricles and the central canal of the spinal cord. This is the ventricular system. The choroid plexus here produces the CSF or cerebrospinal fluid, and then what the layer that wraps around or forms the lining of the ventricles and the central canal is the ependymal cells. And finally, satellite cells are found along the periphery of the neuronal soma or the cell body, and their main function is to provide structure and support the metabolic function of the neuron. Okay, we're going through this again because it's important. So you have your brain, you have your white matter. This is where signal needs to be carried fast. And then you have your gray matter. This is where signal takes time and it gets processed. So the white matter is usually lipid rich and it has many myelin wrapped around it. So you need your oligodendrocytes. While the gray matter usually has less myelin and it's used for processing signal. This is for the spinal cord. So in the brain, the gray matter was on the periphery, while in the spinal cord, it's usually more central. This is the gray matter. And this is the white matter. So here's where you need fast signals. This is where you have more myelin. And the gray matter in the spinal cord can be divided into the ventral horn and the dorsal horn. So the ventral horn is responsible for carrying motor signals from the brain to the peripheral nervous tissue. So the signal is usually going towards the periphery, and then your dorsal horn carries sensory signals towards the brain. So any touch, sensation, vibration, pain is carried through the dorsal horn. And this is the central canal, which is lined by ependymal cells. In order to protect this delicate structure that carries an extremely important function in the body of your meninges, the meninges is made of three layers: your dura, the arachnoid, and the pia. So the pia is in close proximity with the brain, and the dura is in close proximity to the bone and surrounding structures. Now, these are not neuronal tissue or nervous tissue. Instead, they are connective tissue, and they carry the function of protecting the arteries, protecting the brain, and so on. It is also important to understand that they're made of connective tissue. So in case of the dura, it's made of dense, irregular connective tissue. The arachnoid is made of two layers, actually, one which is in contact with the dura and one which is loose. As you can see, these trabeculae or connections are made from the arachnoid space. And then you have your pia, which is in close contact with the brain. And this pia is attached, if you remember, as we said, the glial limiting membrane, which is made of astrocytes. Here we're revising the myelination process again. So the Schwann cell or the oligodendrocyte wraps around the axon multiple times. It removes all the cytoplasm, and this makes the signal able to jump. So this was the myelin. This allows for signal jumping and faster transmission of signals. You can visualize it using an electron microscope. As you can see, these are the different layers of the myelin wrapping, or using a normal light microscope, which shows it as a dense layer around the axon. This is the node of here, and this is where the signal jumps for faster transmission. It's also important to understand that we do have unmyelinated axons, and this exists in the spine and the brain, specifically in the gray matter. On the other hand, peripheral tissues are all myelinated. This picture shows an electron microscope of a Schwann cell wrapping around multiple axons. In this case, these axons have a small diameter, and hence the Schwann cell is able to wrap around them. And it's also important to note that this is not a complete wrapping in multiple layers, but a single wrapping around each axon. And this is the general structure of a spine and the neurons within the spine. So you have your receptors that carry vibration, that carry pain, that carry sensation through the dorsal ramus and then to the dorsal column and attached to the dorsal part of the cranial matter, and then carries it up to the brain. And then you have signals which come from the brain attached to the ventral part of the gray matter and then carry it to the muscle. And then you have your ganglion. A ganglion is basically where the cell bodies of sensory neurons meet outside the central nervous system. In this case, if you go back, so this is a ganglion, this is a dorsal ganglion, and it's where the cell bodies, as you can see here, they meet outside the central nervous system. The peripheral nerve has a specific structure, which starts by an axon and a Schwann cell, which are wrapped around an endoneurium. Now, this structure, which is wrapped around by an endoneurium, is combined with other structures wrapped around by an endoneurium. And these structures are called fascicles. These fascicles are grouped together and wrapped around by a perineurium. And then this all is wrapped around in an epineurium. Now, when it comes to the endoneurium or perineurium or epineurium, this is connective tissue, not nervous tissue. This diagram is nice and it shows that. As you can see here, the endoneurium is made of reticular cells, fibroblast cells, and capillaries. These are all part of the connective tissue, and their main purpose is to protect these precious fascicles. Thank you for joining us today. If you have any questions, feel free to email me.